Welcome to the sixth class of Immunology. In this class, we will discuss about cytokines. As you know, and what we learned in our last class was that cytokines, they are the chemical messengers of the immune system. The kind of cytokines are produced, the different cytokines which work in combination, and their respective receptors, they all decide what kind of immunological response will be generated. So cytokines are very important because they ultimately decide the fate of any immune response. They decide which cells will be activated. They decide which cells will be differentiated. They decide that which cells will be dead by apoptosis. Okay, so cytokines are very important, and actually they are the whole controller of the immune system, right? Because their expression and the expression of their receptors, if you just remove these two, then the immune system will not work. No immune response will be generated. No cell will be activated. No cell will be differentiated. No cell will kill any pathogen. Okay, so cytokines are the main actors apart from the immunological cells which decide the kind of immune response going to take place when any pathogen enters the body. Okay, now let us start this class by the previous class recap. So in the previous class we studied molecular signaling. We studied the important concepts of immunosignaling, how immunosignaling is important, and what are the key concepts, what are the key principles of immunosignaling. Then we discussed about the receptor ligand interaction, which is the key to any signal transduction process. Then we studied the fundamentals of signal transduction, how signal transduction takes place, starting from binding the ligand to the receptor, and then the downstream processes till the transcription factor is generated, and then it binds to the DNA, starts transcription, and then the formation of any enzyme or any cytokine. Okay, then we also discussed the three important pathways of immune system: the PLC pathway, the RAS MAP kinase pathway, and NF kappa B pathway. Okay, so now we will be moving forward with the discussion of the cytokines which play all these pathways and which are the major players of the immune systems in terms of providing communication. Okay, so cytokines, they are small proteins, and usually the size of the cytokines are either equal to or less than 30 kilodaltons. So they are very small proteins, and they communicate among cells of the immune system. So all the communication network, whatever is there for the immune system, that is mediated by cytokines. Now they are either soluble or they can be membrane bound. They are available in both forms, and they bind to its receptor on the target cell. So here you can see that this is the target cell, which is the cytokine producing cell, and here you have the stimulus, and when the cell is stimulated it produces the cytokines, and these cytokines, they bind to the target cell, right? And here you can see the receptor, and on the receptor the cytokine is bound, and then there is a downstream processing, and then up to nucleus level, right, on the DNA, and then the biological effect takes place, it is generated. Okay, so it allows its sufficient binding by changing the expression of cell adhesion molecules and receptors. So these are important because when cell adhesion molecules and chemokine receptors or cytokine receptors, they are expressed in sufficient quantity, then only a particular response is generated. And these can signal an immune cell to either increase or decrease the activity of an enzyme. So basically, they instruct the cells whether to increase or to decrease the activity of a particular enzyme or a particular end product. Okay. Now, the cytokine cells also instruct a cell when to survive and when to die. So basically, their survival and their death is also controlled by cytokines. So when the cell is required, then the cytokines will give the survival signals to the cell. And when the job is over and then the cell is not required, then the cytokines provide the death signal to the cells so that the cells can undergo apoptosis or other kind of cell death. Okay. Now, we also have the chemokines, which are the cytokines that mobilize immune cells from one organ or from one part of an organ to another. So basically, they are the drivers. They drive the cells from one place to another by by the use of their concentration gradient. So here you can see this is the cell which is just going in one direction, and then it can move with the greater speed or it can change the direction depending on the concentration of the these chemokines. And basic one of the classes of chemokines are the chemoattractants. They function by manipulating cytoskeletal proteins and cell adhesion molecules to drive the cellular movement. So you know that the cells undergo changes in the cytoskeletal proteins, and these cytoskeletal proteins are important for the cellular movement. I'm sure you must have learned this in the molecular biology. Okay. Now these cytoskeletal proteins, they are manipulated by these chemoattractants, as well as the cell adhesion molecules are also manipulated so that either they are increased or they are decreased. If they are decreased, then cell is more motile, and if they are increased, then cell is likely to adhere to the surface or to a particular tissue. So chemokines, or particularly the chemoattractant class, it can drive the cells whether to move or whether to stay in a particular environment. All right. Now, there are other cytokines which we call them as the interleukins, and they are the cytokines which communicate between leukocytes, okay, or the WBCs, white blood cells. So, as the name itself is suggesting, between is like inter and leukins are the leukocytes. So, interleukins are the cytokines which communicate between leukocytes, and their numbering reflects their order of discovery. So if there is IL1, IL2, IL3, IL4, so the number 1, 2, 3, and 4, they are basically reflecting their order of discovery. So IL1 was discovered earlier than IL2, and IL2 was discovered earlier than IL3, but after IL1, and so on. Okay. Some cytokines still retain their early names. For example, TNF, for example, interferons, etc. But they are actually ILs, interleukins. So they are ILs, but still, because traditionally they have been named like TNF and interferon, and they are known by these names, so their names are retained. Most immune system cytokines exhibit autocrine, paracrine, and endocrine actions. Okay, so cytokines, they act by autocrine action, by paracrine action, and endocrine action. So here you can see the endocrine action where the cytokine is released, then it goes into the circulation, and then it acts on the distant cell, just like hormones. All right, so these are the endocrine action. Now, in case of the paracrine, the cell secretes the cytokine, and this cytokine, it acts on the nearby cell, okay, within the neighborhood. So this is known as the paracrine action. And in case of autocrine action, the cell generates the cytokine, and the cytokine, it's binding to its the receptor of on the cell itself and then acting. This is known as the autocrine action. Okay. Now, so you know the difference between the endocrine action, paracrine action, and autocrine action. Now, there are membrane receptors which determine the sensitivity of a target cell to a cytokine. So whether a particular cytokine will impact or will affect a particular cell or not, that will be decided by its receptors, right? So very high affinity for receptors and secretion in close proximity, it mediate powerful biological effect. So if receptors are less, the cytokine will either it will have negligible effect or it will just give a very feeble effect. But if the receptors are in high number, then the cytokine will definitely act strongly and will give a profound effect, okay, a powerful biological effect. Now let us study some properties of cytokines. Cytokines, they activate, proliferate, differentiate target cells. Okay. Now here you can see this is the inducing stimulus, and here this is the cytokine producing cell, and the cytokine genes are expressed by this stimulus, and then a cytokines, some classes are secreted, then they bind to the cytokine receptors upon on the target cell, and then again through the downstream processing they do the gene or enzyme activation, and then those genes or those enzymes, they are either helpful in the proliferation, differentiation, or in the cell death of their target cells. Okay. Now, by triggering signal transduction pathways which alter or that alter the target gene expression. So how they activate, proliferate, and differentiate target cells? They trigger a signal transduction pathway, and this signal transduction pathway alter the target gene expression. Now, they also do by regulating the intensity and duration of the immune response. It is important point. They regulate what will be the intensity of the immune response as well as what is the duration or what will be the duration of the immune response. In this way, they will activate, proliferate, and differentiate the target cells. Then how do they do? They do by regulating the secretion of other cytokines or of the antibodies. That is one way. They regulate the secretion of the other cytokines or of an antibody. And those cytokines or antibodies, they define the intensity and duration of the immune response, or by inducing apoptosis in the target cell. So if the cell is not there, then definitely its effect will also be not there. So that's how they regulate the intensity and duration of the immune response. Okay. Now they can also modulate the expression of various cell surface receptors for chemokines, other cytokines, or even for themselves. So modulation means that they are altering or they can regulate the expression of various cell surface receptors for chemokines, other cytokines, and even for themselves. So if the cell surface receptors are available, then definitely there will be binding, and the binding will be there, then signal transduction will be there, and the result is that the production of a particular enzyme or particular cytokine or for particular product which will be used for a particular event, right? So they can modulate the expression of various cell surface receptors. So whether the cell surface receptors will be expressed in low quantity or in intermediate quantity or in higher quantity, they can modulate the expression. And these receptors are for different chemokines or for different cytokines and for even themselves also, like in case of the autocrine signaling. Okay. So that's how they regulate or they modulate the entire event of the immune response. Now these cytokines, they exhibit certain properties, they exhibit certain attributes. What are these attributes? First is the pleiotropy. A pleiotropy means that a cytokine induces different biological effects depending on the target cells. What does it mean? That means that the cytokine remains the same, but on different cells, its effect is different. Now let's understand by this example. This is the activated Th cells. All right. And this activated Th cells, it is secreting IL-4. IL-4 is kind of cytokine. Now this IL-4, when binds to the receptors on the B cell, it causes the B cell to activate, to proliferate, and to differentiate. That is the effect of the IL-4 on B cells. The same IL-4, when binds to its receptor on the TC cell precursor, then it actually causes the TC cell precursor to proliferate and differentiate, but not activate. Okay, so on this, if a cell, the activation part is gone, activate, it cannot activate, while on B cell it can activate. Now when the same cytokine IL-4, it binds on the mast cell, then it causes the mast cell to proliferate but not to activate or to differentiate. So the cytokine IL-4 is the same, okay, but the three different target cells having the receptors for IL-4, they are showing the different effects. So this is known as pleiotropy. So pleiotropy is when a cytokine induces different biological effects depending on the target cells, right? Now the second property is the redundancy. What is redundancy? When two or more cytokines, they mediate the similar functions. There are some cytokines which mediate the similar functions. This is known as redundancy. Now here you can see the activated Th cells, it secretes IL-2, IL-4 and IL-5, right? And all these three, IL-2, IL-4 and IL-5, when they bind on B cells, they help in the proliferation. Okay, so they are three different cytokines, but they are doing the same function on the B cell, which is the proliferation. So this is known as the redundancy. So cytokines exhibit pleiotropy, cytokines exhibit redundancy. Now third is the synergism. What is synergism? When the combined effect of two cytokines on a cellular activity is greater than the additive effects of the individual cytokines. This is known as the synergism. So here you can see this is the activated T helper cell, and IL-4 and IL-5, they are together inducing a class switch to IgE on the B cell, right? So this is known as the synergism, when two cytokines, they show the greater combined effect, okay, than the individual, right? So this is known as synergism. Now next is the antagonism. What is antagonism? It is the effects of one cytokine which inhibit the effects of another. So they work in opposition, right? So this is known as antagonism. Now here you can see the activated Th cells, they secrete IL-4, right? And this IL-4, it causes the B cells, right, to IL-4 causes the B cells to activate, proliferate and differentiate. Here you can see this is the IL-4, but the IFN gamma, it blocks, right, it blocks the activity of the IL-4. So it also blocks the class switching induced by the IL-4, right? So here you can see in synergy, this is IL-4 and IL-4 is causing class switch to IgE. So it not only blocks the class switch to IgE, which is induced by IL-4, but also it also blocks the different, activation, proliferation and the differentiation part to some extent. But this activity is very well observed. The class switching blocking is very well observed as compared to the three activities, right? Now then we have the cascade induction. Now cascade induction is where the action of one cytokine on a target cell, it induces that cell to produce one or more additional cytokines. Now here you can understand the cascade induction. This is the activated T helper cell. This T helper cell is producing IFN gamma. This IFN gamma is binding on the macrophages and then it is causing the macrophage to produce IL-12, right? And then IL-12 is binding to the activated T helper cell and then it is allowing the cells to secrete IFN gamma, TNF, IL-12 and other cytokines. So this is known as the cascade induction. So in cascade induction, what is happening? The action of one cytokine on the target cell is actually inducing the cell to produce another set of cytokine. So here IFN gamma is causing IL-12 production by macrophages, and then IL-12 is causing IFN gamma, TNF, IL-12 and other cytokines production by activated T helper cells. So this is known as the cascade induction. Okay. So these are the different attributes which are exhibited by cytokines, right? Now, the major cytokine producers are T helper cells, dendritic cells, and macrophages. They are the major cytokine producers. Now, the cytokines released from these cells, they are capable of activating entire network of interacting cells. Although these three cells, they are the major producers, but then other cells, here you can see we have the macrophages, we have the B cells, we have the neutrophils, we have here basophils, we have eosinophils, we have T cells, here we have mast cells. It has actually, activated the entire network, and you can see then the different cells, they are communicating to each other using different set of cytokines. So this is activated by the cytokines which are produced by the T helper cells, DCs and macrophages, which are the major cytokine producers. Okay. Now cytokines, they have numerous biological functions. A target cell is exposed to a mixture of cytokines, because it is not just one cytokine when a target cell is exposed. Rather, in a milieu, you will find so many cytokines which are there in the environment, but they will bind to the cells depending on the expressed receptors on the cell, and when different receptors are expressed, then different cytokines, they bind to the cells. So a target cell is exposed to a mixture of cytokines. Now their combined effect can have a wide variety of consequences, and that's how the cell, it fine tunes its own function depending on different kind of cytokines which are bound to the cells, their combined effect, and then which cytokine has bound with the greater affinity, which cytokine has bound with lesser affinity, and what are the different their synergism and antagonism. All right, so that's how the cell decides what is the outcome of a particular impact by these different cytokines. Okay. Cytokines can stimulate and activate T cell subpopulations. Now how? You can see here the IL-12 and IFN gamma. These are the two cytokines. They induce the differentiation of Th1 cellular subset. Okay. Now when Th1 cell is induced by IL-12 and IFN gamma, then it causes the differentiation and activity of the macrophages and T cytotoxic cells, right? And together they cause the cytotoxic immune response. All right, these cells cause the cytotoxic immune response. Now, if you see IL-4 and IL-5, they induce the differentiation of Th2 subset, and when they secrete the Th2 cells secrete the cytokines, they cause the activation of the B cell, and here the effect is the antibody or complement activation. Okay. Now IL-17 and IL-23, they induce the differentiation of Th17 cells, and then Th17 cells, they produce the cytokines which cause the differentiation and activity of macrophages as well as for neutrophils. So they are involved in the inflammatory state. So they are producing inflammation. So you can see that cytokines can stimulate and activate the different T cell subpopulations. So here are the examples of Th1 cell, Th2 cell and Th17 cell. and Th1 cell is causing cytotoxic immune responses, Th2 cell causing antibodies and complement activation, while Th17 cell is causing the inflammatory state or inflammation. Okay. Cell activation may alter the expression of receptors and cell adhesion molecules. So cytokines are concentrated between secreting and the target cells. Okay, they are concentrated where? Between the secreting and the target cells. A very short half life, so act only for a limited time and over a short distance. Now this is how the immune response is controlled, because cytokines, they have a very short half life. Their own half life is very short, and therefore they act only for limited time and over a short distance. So here you can see this is the Th cell, right? And it is involved with the B cell with using the here the antigenic peptide and the CD40 and CD154 co-receptors, right? Now you can see here the cytokine produced by the cell, it is acting upon immediately on the B cell which is already engaged with it. So the cytokines are secreted and they are concentrated between these two cells, and because their half life is short, so they are acting on only for a limited time as well as for the short distance, because these two cells are already in engagement. Therefore the cytokine is immediately released here and it is binding to the receptor which on the cell just adjoining in which is in contact with the secretory cell, right? Now signaling through multiple receptors can fine tune a cellular response. I just told you that the signaling through multiple receptors, because multiple receptors are involved, multiple cytokines are involved, so the cell can fine tune the response, what kind of response it will give, and it all depends what kind of response is required also, okay, by our immune cells. So the actual response mounted by a cell to a signal, it depends upon the nature of the individual receptor and all of the downstream adapters and enzymes present in the recipient cells. So you know that what is the nature of the individual receptor, we can just understand this point, because the one receptor, it will bind to its specific cytokine. So the nature of the individual receptor, moreover the receptor is whether of low affinity, of intermediate affinity, or of high affinity. So that decides the nature of the individual receptor. And all the downstream adapters means all the proteins which are present in the cytoplasm and which are activated by binding to their the cytoplasmic tail of the receptor, and then using phosphorylation, dephosphorylation events, and then they activate the further proteins or the further downstream mediators which are present in the cytoplasm, and then they will go finally to the nucleus. So they are the adapters and the enzymes which are present in the recipient cells. So many are glycosylated for stability. So many cytokines, they are glycosylated, that means they have the sugar residues, and that provides them the stability. So here you can see this is the human IL-7 and this is the glycosylation part. So you can see this glycosylation part here. So it is glycosylated, and this glycosylation is actually providing the stability, the structural stability of this IL-7 or all of the cytokines. Now there are 6 cytokine families, and these are IL-1. These are the members which are important for inflammation, right? And examples are IL-1 alpha, IL-1 beta, IL-1 RA, IL-1 18, and IL-33. The second family is class 1, which we also call as a hematopoietin cytokine family or class 1 family, And the members of this family, they display sequence and functional diversity. So it's a very, very diverse family. So you can see IL 2, 3, 4, 5, 6, 7, 12, 13, 15, 21, 23. Then GM-CSF, which is the granulocyte monocyte CSF. Then GCSF, this is the granulocyte colony stimulating factor CSF. Then we have the growth hormones, prolactin, erythropoietin, hematopoietin. So you can see, it's a very, very diverse family. Okay, as well as it, because it is diverse, the functional diversity will be evident. Now then we have the class 2, and this class 2 family is also known as the interferon cytokine family. So we have the interferons here, which have the important role in antiviral responses. So we put all the interferons in class 2 family. Then we have the TNF family, and this TNF is the tumor necrosis factor family, and their members are involved in the immune system development, effector functions, and homeostasis. So they have a lot of functions here. A variety of functions are seen here in this TNF family. Okay. Then we have the fifth one is the IL-17. So IL-17, we have IL-17A, IL-17B, C, D, E, and F. And the members function to promote neutrophil accumulation and activation, and they are pro-inflammatory. So basically they are involved in the inflammation. And the sixth family is the chemokine family, and all members, they have the chemoattractant functions. They have the chemoattractant functions. Okay, so the, as well as the chemo repulsive function also. So we will talk in detail when we will study this chemokines, okay, after few slides. So let us focus on the IL-1 family, and let us see what are the different properties of these families and how the signal transduction takes place. So IL-1 family, they are very early secretions. So IL-1s are secreted in a very early phase by DCs, which are the dendritic cells, monocytes, macrophages, following recognition and innate receptors. So when innate receptors, they are, so what are the innate receptors? You remember the PRRs, and PRRs are what? PRRs are the receptors which bind to the PAMs or DAMs, right? Pathogen associated molecular patterns, and PRRs are pattern recognition receptors, which are present on the DCs, monocytes, macrophages, and almost all the immune cells there. And they are mainly used for the innate responses. So we call them as the innate receptors. Okay. So once the bacteria or virus or their components, they bind to the PRRs which are present on the DCs, monocytes, and macrophages, then they secrete IL-1, members of the IL-1 family, right? So very early secretion in immune response. Now the uniqueness is that the IL-1 family and TLR, which are the toll-like receptors. Now toll-like receptors are the innate receptors. Okay, toll-like receptors are the innate receptors. So IL-1 family and toll-like receptor families, they share similar function. Okay, so their function are almost similar, right? So this is a very unique thing for in this IL-1 family, primarily associated with innate immunity. Okay, because they are involved with the PRRs and they are involved with the PAMs and DAMs. So they are associated with primarily with the innate immunity. And many biological properties of this family are non-specific, like innate. And you remember that the innate immunity is non-specific, right? So many biological properties of IL-1 family or interleukin 1 family are non-specific, very much like the innate immunity. Members interact with dimeric receptors to induce pro-inflammatory response. So their response is pro-inflammatory and they interact with the dimeric receptors. Okay. Now members trigger innate inflammation via IL-1 family of receptors, while TLRs trigger inflammation via PAMs, microbial products, viruses, nucleic acids, and DAMs. Now you know that they are unique. The IL-1 family and TLR families share similar functions. So their ligands are different. So here in case of IL-1 family, right? So IL-1 receptors are involved because IL-1 is what cytokine and they need the receptor which is the IL-1 receptors. While in case of TLRs, the triggers are, so their ligands are basically PAMs, microbial products, viruses, nucleic acids and danger associated molecular patterns. Okay. IL-1 alpha is a dual function cytokine. IL-1 alpha is a dual function cytokine and it is found in the nucleus where they act as DNA binding protein. The same cytokine binds to the cell membrane receptors and initiates signal transduction. Now that's why we call it as a dual function cytokine. It is found in the nucleus and it acts as a DNA binding protein. While the same IL-1 alpha, it also binds to the cell membrane receptor and it initiates a signal transduction. So it has a dual role. Okay. Now let us see the signaling of the IL-1 family here. So IL-1 binding, you can see here this is IL-1. So this IL-1 binding to the receptor, this is IL-1R1. This is receptor. Okay, R, wherever you will find R, always remember it is receptor. Wherever you will find L, when we do write cytokines, you always remember that it is a ligand. So this is IL-1 and here it is IL-1R1. So this is receptor. So IL-1 binding to its receptor, it causes the conformational change in the receptor's tall IL-1R or TIR domain. So here you can see the TIR domains, which are the cytoplasmic domains. Okay. Now these cytoplasmic domains, they undergo conformational changes when the IL-1 binds to the receptor. Now this allows binding of the adapter protein which is MyD88 via its TIR domain. So here you can see this is the adapter protein MyD88. It immediately binds to the TIR domain once there is a change in the conformation by binding with the IL-1. Okay. Now this MyD88 recruits IL-1 receptor activated kinases which is IRAKs. Here you can see it has recruited IRAKs which phosphorylate one another, facilitate binding of TRAF6. So they will phosphorylate one another and then this TRAF6 it will bind here because due to the phosphate group here. The IRAK-TRAF6 complex, it dissociates and interacts with TAC1 and its two binding proteins TAPS1 and 2. So here you can see this is TAC1, this is TAP1 and this is TAP2, right? Now, TRAF6 and ubiquitin ligase, they catalyzes the generation of polyubiquitin chains activating the TAC1 complex. So this TAC1 complex will be activated. How? TRAF6 and ubiquitin ligase, they generate polyubiquitin chains. So they will generate the polyubiquitin chains and then this TAC1 complex will be activated. So TAC1 activates transcription factor NF-kappa B, also activates MAP kinase cascade to activate AP1 transcription factor. So here you can see this, here you can see the activation of NF-B pathway and here you can see the MAP kinase cascade. So ultimately we are uh, here in the nucleus where AP1 transcription factor complex and NF-kappa B, they go and they bind to the DNA and then they start the expression of the specific cytokines, and that's how the IL-1 family of proteins, they act on a particular cell and then they start the signal transduction process. Now, the second family is the hematopoietin family, which we call it as a class 1 family. So when we say class 1, don't think it is IL-1. IL-1 is different, class 1 is different. So class 1 is the second family, which is the hematopoietin family. And this is the largest family. It has small soluble cytokines which exhibit diversity of functions and not all members have the hematopoietic functions. right? But the family is called hematopoietic family or hematopoietin family because of the earlier members which were discovered and have been found in the hematopoiesis. Okay, but not all members have the hematopoietic function. So they have the 4 helix bundle which is organized into 4 anti-parallel helices which define the structural feature of this family. So here you can see now this is the N terminal, okay, and you start with the N terminal. So this is A. You can see it's a yeah, the helix A, right? And then here you can see it is coming to helix B, and from here B to C, and from C we are going and going to D, and then we are reaching to the C terminal domain or C. All right. So here you can see the D, A, C, and B, they are all anti-parallel. Means their direction is opposite. So D is opposite to A. You can see the direction of the arrow. The direction of A is opposite to C. You can see the change in the arrow here. It's going forward, it is going downward. And then C and B are also anti-parallel. So we call this kind of situation as an anti-parallel. So these helices are anti-parallel and they are 4 helix bundles. So here you can see A, B, C and D, and they define the structural feature of this family. So all the family members, they will have this structural feature. All right. Members subclassification is the basis is the helical length. So IL2, 4, 3, they have the short chains around 8 to 10 residues, while 6 and 12 they have 10 to 20 residues. So further the family is subclassified based on the helical length, short length and long length. So IL2 begins B or T cell proliferation. Okay, now these are some of the functions. IL2 begins B and T cell proliferation. IL4, it regulates the T helper function. IL6 involved in B cell differentiation. IL4 and IL12, they are involved in the differentiation of T helper cells, and granulocyte monocyte colony stimulating factors or granulocyte colony stimulating factor, they are involved in the WBC differentiation or leukocyte differentiation, right? So most class 1 cytokine receptors, now we are talking about the cytokine receptors, okay, class 1 receptors. We talked about the cytokines, so here we are talking about their receptors. So most class 1 cytokine receptors, they have multiple subunits. Subunit beta, GP130 or gamma C. Okay, they define 3 subfamilies. Each subunit can pair with different alpha chains forming specific receptors, right? So these are the 3 different subunits and their partner chain will be the alpha chain and they form the specific receptors. So alpha subunit, it mediates cytokine binding and partner chain mostly cause a receptor signaling. So beta, GP130 and gamma, they contribute to cytokine affinity, right? So here you can see this is the alpha chain and then they are the partner chain beta, and you can see the alpha and beta, they form, if they are together, they form the high affinity receptor. But if alpha chain alone, although it can bind with IL3, but it binds with a very low affinity. Here it can bind IL5 or GM-CSF, but the affinity is very, very low unless the beta chain also binds and it gives a dimeric situation where the affinity becomes high. Now, there is one disease which is known as X-linked severe combined immunodeficiency disease, which is XSID. It is because of the defect in the gamma chain gene. So here you can see the receptor is the gamma subunit. When there is a defect in this, then we see this X-linked severe combined immunodeficiency disease. Okay. Now let's move to the third interferon family, which is the third cytokine family, which is the interferon family or class 2 family. There are three subfamilies of the interferon. We have type 1 interferon, we have type 2 interferons or IFN gamma, and then we have type 3 interferons, which is IFN lambda. Okay, and they are very potent antiviral cytokines. Remember, they are antiviral cytokines. IFNs are antiviral cytokines. In type 1 family, we have IFN alpha and IFN beta subfamilies. So IFN alpha and IFN beta in type 1. Then in type 2, we have IFN gamma. And in type 3, we have an IFN lambda. So members of both dimeric helical proteins of around 18 to 20 kilodaltons, and they are secreted by activated macrophages, dendritic cells, and PRR mediated recognition of viral infected cells. Okay, so when a cell is virally infected and then it is recognized by the PRRs, so that's those cells, macrophages and DCs, they secrete this IFNs because they are antiviral cytokines. They induce ribonuclease production, and these ribonucleases, they can degrade the viral RNA. Okay, so type 2 IFNs, which we are IFN gamma, they are produced by activated T and natural killer cells. So IFN gamma is secreted by or produced by activated T and natural killer cells. They induces macrophages, they stimulate TC differentiation. IFN gamma induce macrophages as well as stimulate the T cytotoxic differentiation, and they are made by Th1 cells which support cell mediated immunity. Okay, so basically they are supporting the cell mediated immunity. Inappropriate synthesis, they stimulate DTH, resulting in the tissue damage. All right, so tissue damage is one of the inappropriate synthesis causing event, and then it has to be in a very optimum concentration, otherwise the result will be the tissue damage. IL-10 is secreted by monocytes, T cells, B cells, and dendritic cells, and they regulate immune responses. Okay, now if we see the type 3 IFNs, which is the IFN lambda, it induces special dendritic cells, and these cells are called plasmacytoid dendritic cells. And these plasmacytoid dendritic cells, which are special dendritic cells, they secrete type 1 interferons. Okay. They are critical against pathogens at epithelial barrier surface. You remember epithelial barrier surface? We have the skins and mucosa. Okay, they are the epithelial barrier surface, and these special dendritic cells, which we call plasmacytoid dendritic cells, they are very critical against the pathogens which are at the interface here at the epithelial barrier surfaces. They have effects on innate and adaptive immunity, and they can promote inflammation in certain conditions. So the class 2 cytokine receptor family. Now we are talking about the receptors. We talked about the class 2 interferons. Now we are talking about the class 2 receptors. So class 2 cytokine receptor family has 12 receptor chains that can rearrange for binding more than 25 different class 2 cytokines. So just 12 receptor chains, they will rearrange themselves, and they will rearrange in such a way that they can bind to more than 25 different class 2 cytokines. So that's how the power of rearrangement in receptors, although they have very limited sets, but by rearranging and by reorganizing themselves and by pairing with different partners, they can increase their diversity to bind the different type of cytokines. Now let us understand some clinical therapy with interferons, and interferons are used for a number of conditions. So IFN alpha, it is used to treat hepatitis B and C. hairy cell leukemia, chronic myelogenous leukemia, and Kaposi's sarcoma. So basically, these are three cancer conditions, and this is virus. IFN gamma, it is used in multiple sclerosis, okay, and IFN beta is used in multiple sclerosis, and IFN gamma is used in non-Hodgkin's lymphoma, cutaneous T-cell lymphoma, multiple myeloma, chronic granulomatous disease, and osteopetrosis. So these are different clinical conditions where IFN therapy has been successfully used to control the clinical conditions, right? Now, if we see the signal transduction model, this is just the signal transduction model, but most class 1 and class 2 receptors, right? Not IL-1, but class 1 and class 2 receptors. So class 1 receptors are what? The hematopoietin family, and class 2 receptors are IFN, right? So here you can see this is the JAK-STAT signaling pathway. We call it as a JAK-STAT signaling pathway. So JAK is Janus activated kinase. So they are JAKs. Okay, that's a short form or the abbreviation of for Janus activated kinases. They are tyrosine kinases, TKs. Some retain their earlier name TYK. The earlier name is TIC. Now, signal transducers and activators of transcriptions are STATs, and these are the transcription factors. So, the abbreviation of signal transducers and activators of transcriptions is STAT, and they are transcription factors. So, JAKs, they are the tyrosine kinases, while STATs are the transcription factors. Clear? Now, cytokine binding associates the two receptor subunits and activates the receptor associated JAKs. So what you can see here, this is the JAK, okay, and these are the receptors alpha and beta chains. But when the cytokine binds, it brings these two chains closer. So you can see here they brings the dimerization. So here it is the dimerization of receptor, all right, and this dimerization, it activates the receptor associated JAKs. So the JAKs phosphorylate specific tyrosines in the receptor subunit. So you can see here, this is the JAK. It will phosphorylate the specific subunits on the receptors, right? The phosphorylated tyrosine residues, they act as a docking sites for inactive transcription factor STATs. So now STATs, they can easily bind here because they provide the docking site, the site where the STATs can bind, and these are the inactive transcription factors. They are not active yet. The inactive STATs are phosphorylated by JAK and TIC kinases. So you can see here that once they bind here, then these STATs are also phosphorylated, and they are phosphorylated by JAK and TIC kinases. Okay. Phosphorylated STAT transcription factors, they dimerize. So here you can see they have dimerized. Okay. Here. So this is the dimerization state, and this is the phosphorylated state, and in opposite structures they have put their phosphate into the grooves of the other stat. So they dimerize binding to one another. Now phosphorylation, it changes conformation in the stat dimer to reveal a nuclear localization signal. So in molecular biology, you must have heard this nuclear localization signal, which actually is the signal allowing the particular protein to enter the nucleus. So in this case, we have the STATs. Okay, so STATs, once they are dimerized, so they give or they reveal a nuclear localization signal, and this nuclear localization signal helps them to enter the nucleus. So the STAT dimer, it translocates into the nucleus where it initiates the transcription. So here you can see it is binding to the DNA to start the transcription, right? So this is a very general model by most class 1 and class 2 receptors, and how their cytokines, different cytokines, they bind, and then they form this stat formation, and this stat causes the DNA to undergo specific gene transcription. Okay. Now, the fourth family is the TNF family, which is the tumor necrosis factor family. So, TNF cytokine family and TNF nerve growth factor family of their cognate receptors. So, this is the cytokine family, and this is the TNF NGF cognate receptor. This is the receptor family. So, all cells in the body, they express receptors, and many also express some ligands of these families, and they assemble into trimers, homo or heterotrimeric. So either all the three units are the same, then they are the homotrimers, or if one or two units are different, then we call it as a heterotrimer. So they assemble in trimers. So their functions are tissue homeostasis and embryonic development, okay, and tissue homeostasis. The functional diversity of this family is due to the distinct features of the members and the interactions with non-family ligands and co-receptors. So they not only bind with the family members, the receptors, but also their interaction with the non-family ligands, the ligands which do not belong to TNF family. So their interactions also provide the functional diversity. Okay, they have a different functions, a diverse set of functions. Now, membrane bound and soluble forms of ligand. So they have the membrane bound as well as the soluble forms of ligand and receptors. So they are free receptors, there are free ligands, there are membrane bound receptors, there are membrane bound ligands. Then re-anchoring of soluble forms to ECM components, re-anchoring, they can anchor again the soluble forms to ECM components. So they are soluble forms, but they can bind to the membrane. So this is known as the re-anchoring. And then signaling initiation via intracellular domains of the receptors and ligands. So these are some factors which provide functional diversity to tumor necrosis factor family. All right. So they bind with their own family members as well as the non-family ligands, their co-receptors. Then the ligands and receptors, they are in soluble form as well as the membrane bound form. Then some soluble receptors or some soluble proteins, they can re-anchor. All right. And then the signal initiation is via intracellular domains, which are the IDs of the receptors and ligands. So all these factors, they contribute to the functional diversity of TNF family proteins. Now TNF alpha, which we call it just TNF, whenever in text you will find TNF, by default it is TNF alpha. Okay, so TNF, it is a pro-inflammatory, that means it increases the inflammation. It is produced by activated macrophages, lymphocytes, fibroblasts, and keratinocytes against infection, inflammation, and stress. Even if there is some stress, whatever the stress is, it can also produce the inflammatory state, right? Apart from the infection and the inflammation, the usual injury or usual, other factors which can cause inflammation. TNF beta is called lymphotoxin alpha or LT alpha. So LT alpha or TNF beta, it is the same name. Okay. So binding to neutrophils, endothelial cells, and osteoclast, and they deliver activation signals. In other cells, the binding can increase expression of MHC and cell adhesion molecules. So basically TNF beta's job is to activate the different cells. So they provide the activation signals, and in some other cells it can also increases the expression of major histocompatibility complex MHCs and cell adhesion molecules. So they have a different functions. Then lymphotoxin beta, okay, it is critical in lymphocyte differentiation. So this cytokine is particularly important for lymphocyte differentiation. And then we have BAFF and APRIL, which are important for B cell development and homeostasis. So these are some of the TNF family members and their different roles. CD40L. Now, I told you that wherever you will find L, that means ligand. So, this is CD40 ligand on T cells. It signals B cell differentiation. So, this ligand, it is present on the T cells, but when T cells they come and close to B cells, they interact with the B cells, then it signals the B cell differentiation. Then we have the fast ligand. So we call it as a fast L or CD95L. It induces apoptosis on binding to its cognate receptors. Okay, fast or CD95. So here its role is to give a death signal, right, by binding. So this is about the TNF family of proteins. Now we have the TNF receptors and we have the receptor superfamily, which is the CRD, cysteine rich domains in the ligand binding domain. So basically on the receptor where the ligand binds, on that particular area we have some domains which are rich in cysteine, and we call them as a cysteine rich domains or CRDs. Now each CRD contains 6 cysteine residues to form 3 disulfide bonded loops, and individual members can have 1 to 6 CRDs. So these are the some structural features of TNF receptors, where the cysteine play important role in forming the 3 disulfide bonded loops by 3 cysteine residues in each CRD, and an individual member can have 1 to 6 CRDs. Okay. Most TNF receptors are type 1 membrane proteins. Okay, their N terminals are outside the cell and few are soluble. So membrane proteins are also classified, which is not the scope of today's discussion, but you can consult any molecular biology book. So this is type 1 membrane protein where their N terminals are outside the cell and few are soluble. Some lack a membrane anchoring domain. Okay, some don't have the membrane anchoring domain and may link to the membrane only by covalently bound glycolipid anchors. So although they lack a membrane anchoring domain, but still they can bind to membrane by covalently bound glycolipid anchors. And these soluble receptors, although they are not membrane bound, they are soluble receptors, but still they can link to membranes using these glycolipid anchors, and these soluble receptors are known as decoy receptors as they are capable of intercepting the signals from the ligand before it can reach a cell, blocking the signal. So their main function is to catch the signal before it can reach a cell, and so that it can block the signal transduction. So these are known as the decoy receptors. Now, interaction of one trimeric TNF molecule with its receptors induce receptor trimerization and activation. So, in this picture, you can see there we have the TNF receptor, and this is not in trimerized form, but once a TNF, which is a trimer, when it will bind to these receptors, it causes the trimerization, and then once it is trimerized, then it is activated. Now you can see that these three uh, components, they can be homo as well as heterotrimeric. Okay, if they all three belong to the same family or the same type of receptor chains, then we call them as a homotrimers. But if one or two are different from other, then we call it as a heterotrimer. Okay. Now, the signaling of TNF family members is through the Fas receptors. So, the Fas receptor trimerizes on interaction with Fas ligand, and it transduces a signal to the Fas bearing cell, and it results in apoptosis. So, apoptosis or programmed cell death, it is a mechanism where the cell dies from within, fragmented into membrane bound vesicles, which are cleared by macrophages. So it does not produce much drama when apoptosis takes place. It silently, it's just it's dead from inside, and then their components are packed in small packets, and there is no much inflammation or no much kind of inflammatory or other thing like in case of necrosis, and then these are cleared by the macrophages. So here you can see there is the cell, then the mild convolution, chromatin compaction, and then cytoplasmic condensation, and then there is a nuclear fragmentation, blabbing, apoptotic bodies are formed, and once these apoptotic bodies are formed which are membrane bound, then they are phagocytosed by the phagocytic cells and the cell is cleared. Okay, so that's how the programmed cell death takes place. So there is a minimal inflammation and the natural end of immune response. So there is not much symptoms which we see in day-to-day apoptotic process. Activation of the apoptotic pathway invokes the activation of caspases. So caspases are basically the proteases. These are the enzymes, and they bear cysteine residues at their active sites, which cleave after aspartic acid residues. So they are the main enzymes which are involved in the apoptosis or apoptotic pathway or programmed cell death. Now here is the signal transduction process of the tumor necrosis factor family. So binding of the Fas L, L is what ligand. So here this is the Fas L. It has bound to the receptors, and then we have the fast death domains. So these are the DDs. Okay, fast death domains. Now DDs of the adapter protein FADD. So we have the adapter protein FADD. It binds to the clustered fast DDs. So we have the clustered fast DDs. So death effector domains, they are located on the FADD, incorporate the DAD domains of procaspase 8 into the membrane complex, right? So you can see here procaspase 8, it is incorporated here and into the membrane here, procaspase 8. Now clustering of procaspase 8, it induces cleavage of their prodomains, releasing the active caspase 8 protease. So you can see here the active caspase 8 is released, okay, from here. Now this caspase 8, it cleaves the pro-domain from caspase 3 and caspase 7. So we have the pro-caspase 3, pro-caspase 7, they are the inactive forms, and it will cleave the pro-domain. Okay, it will cleave the pro-domain and it will convert them into the executioner caspases. Executioner caspases are the caspases which bring about the apoptosis process, which cleaves and activate nucleases, leading to the degradation of the nuclear DNA. So here you can see this is the active caspase 3, this is the caspase 7, which are the executioner caspases, and they cause the inactive apoptotic enzymes into active apoptotic enzymes, and ultimately they are bringing about the cell death. Okay, the programmed cell death. Now caspase 8, it also cleaves and activates the pro-apoptotic BCL2 family member protein BID. So this is also pro-apoptotic, right? Now we see here the signaling through TNF-R1 receptor. So you can see here this is the R1 receptor is in the trimeric form 1, 2 and 3. So TNF-R1 receptor, it is present on the surface of all vertebrate cells, right? And like FAS, it has the intracytoplasmic death domain. So you can see here it has DD, which is the death domain, and intracytoplasmic inside the cytoplasm. It can bind TNF alpha and LT alpha. Okay, so it can bind TNF alpha, LT alpha. So here you can see TNF binding to TNF-R1 receptor, it can lead to two very different outcomes. So once if TNF is binding to TNF-R1, either it will undergo apoptosis, that is one outcome, or other is the survival, which is the life. So here you can see this is the apoptosis, and here it is the survival. It all based on the binding of the TNF to this TNF R1 receptor. Now how it happens, how the cell will decide, or how this the receptors are deciding, or how these whole signal cascade is deciding whether the cell will undergo apoptosis or the survival. Let us check it. So now here you can see in the survival pathway, right? TNF binding, it results in TRADD and RIP1 recruitment. So here you can see they have bound. This is the TNF trimer. This is the TNF R1. It has bound here, and once it has bound, so you can see here it results in this TRADD. This is TRADD and RIP1. This is RIP1, the recruitment. They both bind to the DD here. Now, TRADD recruits CIAP1 and CIAP2, which is the ubiquitin ligases, which bind to Lubac protein. So here you can see Lubac protein, and this is IAP1 and IAP2. Ubiquitinated RIP1, okay, binds to Nemo in IKK complex and TAC1. So this is TAC1 and Nemo IKK complex, not shown here, but it binds there. Okay. Now TAC1 and RIP1, this is TAC1 and RIP1, they activate IKK complex, degrading IKB and releasing NF kappa B. Alright, so here they are releasing NF kappa B, and it enters the nucleus. Pro survival effects, it activates the C flip transcription. So here you can see the C flip transcription takes place, and it causes the survival and activation of the innate and inflammatory genes. Now in apoptotic pathway, right, TNF binding to TNF-R1. So here you can see TNF binding to TNF-R1 induces trimerization and conformational change. Definitely it will happen here. Now this causes the binding of DD containing adapter TRADD to the internal face of TNF-R1. So here you can see the TRAD is bound to internal R1. TRAD binds to RIP1, which is a serine threonine kinase via its own DDs, and TRAF2. This is complex 1. So here it is TRAF2, this is RIP1, and this is TRAD, and we call it as a complex 1, right? There is no CIAP2 or Lubax, as was the case in survival pathway. So complex 1, it detaches from the TNFR, okay, from the receptor. This complex 1 is detaches and migrates to the cytoplasm to bind FADD. So here you can see this whole complex 1, it detaches itself and then it binds to this pink color FADD, right? And FADD recruits procaspase 8 driving apoptosis. So this procaspase 8, which is in inactive state, it becomes caspase 8. It is active, and it blocks the C flip, which causes the survival and activation, and it allows the cells to undergo apoptosis. So that's how the same receptor, it can either drive the cellular part towards survival or towards apoptosis based on the different bindings here. So the survival versus death decision, it depend upon the outcome of the race between the generation of active caspase 8 on one hand and the caspase 8 inhibitor C flip on the other hand. So C flip can actually blocks the caspase 8. So if C flip is formed, then caspase 8 is blocked and the cell will undergo survival. But if C flip is not formed, so the caspase 8, which is which will cause the apoptosis. So it all depends, whether we have the C flip or not. So if C flip is generated, then the cell will survive. Why? Because it will block the caspase 8. But if it is not generated, then the cell will undergo apoptosis. Okay. Now the fifth family is IL-17 family. So IL-17 is the most recently identified cytokines. We have 6 members in this family: IL-17A to IL-17F, A, B, C, D, E, F. Total 6 members. So members have various biological functions. For example, inflammation during infection, autoimmune disease, as well as boosting protective immunity against various pathogens. So members share a highly conserved, tightly folded dimeric structure with 4 conserved cysteines and usually found at the interface of innate and adaptive immunity. Interface of innate and adaptive immunity. They serve to coordinate the release of pro-inflammatory and neutrophil mobilizing cytokines, right? So IL-17A is a very highly versatile pro-inflammatory cytokine which causes inflammation or which helps in inflammation. It is released by activated Th cells and its receptors are found on neutrophils, on keratinocytes and other non-lymphoid cells. The T cells secreting IL-17A are the Th17 cell subset. I am sure you must have learned this and you must remember this when we discuss the T helper subsets, during the course of our earlier discussion. IL-17E, this 17E, it suppresses Th17 cell response. So basically it is the suppressor of Th17 cellular response. And this IL-17 receptor family, it is composed of 5 protein chains. It has 17RA, so receptor A, RB, receptor B, RC, receptor C, RD, receptor D, and RE, receptor E. So we have the 5 protein chain families of receptors, okay, R, A, B, C, D, and E. They are arranged into homo and heterodimeric and trimeric units to form the complete receptor molecule. So a receptor molecule consists of uh, dimeric or trimeric units, and these units can be homo or hetero based on the composition of the receptor molecules. So here you can see this is the trimeric unit. Okay, so this is 2 or this is IL-17A, right? You can see here this is again the heterodimer. This is 17B and 17A. This is homodimer 17RB. Both chains are of RB. This is also homodimer 17RE. This is unknown receptor. So this is also dimer 17RD, and this is heterodimer RD and RA. So these chains can easily change, or they can reorganize themselves to form a different receptors. So they can bind to different type of 17 members: IL-17F, A. Here you can see IL-17E, B, C, D, unknown ligand, but yes, we know the receptors here. Okay, so that's how the receptors and their cytokines, they are arranged and they can bound to each other. Now, family shares fibronectin domains. So, this receptor family they have a fibronectin domains with the hematopoietin and interferon family cytokine receptors and are single transmembrane proteins. So, they are the single transmembrane proteins and they share the fibronectin domains which are found in hematopoietin and interferon family cytokine receptors. So, family receptors of hematopoietin, receptors of interferons and receptors of IL-17, they all have this fibronectin domain. They share this structure, right? Now, if you can see here, IL-17, it does a lot of work in terms of host defense against pathogens, like it is against fungal infection, bacterial infection, and as a mucosal barrier. But side by side, it is also causing a number of disease, and it is known to cause psoriasis, it is known to cause psoriatic arthritis, and it is known to cause ankylosing spondylitis. While there are some questions about its role in tumors, IBD, and autoimmune disease. So, if we can block this pathway which can lead to these diseases, then probably we can find some new drugs. All right, so most of the new drugs against psoriasis, psoriatic arthritis, and ankylosing spondylitis, they are being developed on the basis of their role in inducing these diseases. Okay. Okay, now the fifth family is IL-17 family, and this IL-17 family transduction, which we were just talking about, and we have started talking about a few slides back. Now you can see here the transduction pathway process here. Like IL-1 receptors, the signaling is through most IL-17 receptors, and it results in the inflammatory response. It results in the activation of NF-kappa B, which is a hallmark transcription factor of inflammation, right? So you can see here this is IL-17R, okay, and then these are the their cytoplasmic domains, and then there are various pathways which are going and causing NF kappa B activation, then MAP kinase cascade and mRNA stabilization, and this is causing NF kappa B, the cytokine expression, and the CEBPs cause IL-6 expression. So IL-17 receptor binding recruits adapter protein ACT1. So you can see here this is ACT1 protein which is recruited here, and it serves as a docking point for TRF protein 3 and 6. So we have TRFs. So TRF3, TRF6, they bind to ACT1. They recruit members of the TAC1 complex. So this is the TAC1 complex, and TAC1 activation results in the activation of IKK complex and NF kappa B activation. Okay, so it is not shown here, but we have in many transduction processes in our earlier discussions we have shown this process. So they activate the IKK complex and then NF kappa B activation. Now, in the second part, the adapter protein bound to Saphir domain. This is Saphir domain, and it recruit MAP kinase pathway component. So basically, this domain is for MAP kinase pathway components. So the MAP kinase ERK1, it phosphorylates cytoplasmic protein uh, triacetylproline, which is mRNA degrading protein, and inhibits its ability to bind to AU-rich elements on mRNA, which encoding cytokines. All right. So basically, if you can see that the CSIR is the key for the MAP kinase pathway, and this MAP kinase component, which is the ERK1, right, this is also important, and everything is recruited here after one another, and they do the whole downstream activation of one protein and another, and then finally they activate this MAP kinase pathway. All right. Now, activation of this pathway results in enhancing the stability of cytokine messenger RNA, and this cytokine messenger RNA is stabilized. So you can see here, so it is already stabilized. All right. And this stabilization is important. Now, IL-17 receptor binding also results in the activation of transcription factor, which is CEBP family. So you can see here CEBP family here, and it promotes the expression of the inflammatory cytokine IL-6. So IL-6 will be expressed here. Okay. So you can see here that when IL-17 binds to the IL-17 receptor, so the outcome can be different. Outcome can be NF-kappa B activation, outcome can be the stabilization of messenger RNA, and outcome can be the expression of the IL-6, depending on what kind of adapter proteins and what kind of downstream proteins are activated, or further they are activating the other proteins, and then how the pathway is going. So you this is a very good example where one receptor, it can bind and it can activate different outcomes. Now, the last family is the chemokine family, and this chemokine family, basically, the members are structurally related, small, low molecular weight cytokines, and their molecular weight is around 7.5 to 12.5 kilodalton, very small. Okay, they bind to cell surface receptors, and they signal through cell surface G protein coupled heptahelical chemokine receptors. So G protein coupled. So basically they are GPCRs, and they are heptahelical. Hepta means 7, and there are 7 helix in the receptor, chemokine receptors. So members share 2 or 4 conserved cysteine residues at particular points in their sequence that form intra-chain disulfide bonds. Now, a variety of directed and undirected migratory behaviors are shown, like chemotaxis, haptotaxis, chemokinesis, and haptokinesis, in addition to inducing cell arrest or adhesion. The primary amino acid sequence and arrangement of specific structurally important cysteine residues, they determine the 4 subfamilies. So basically, if you see the chemokine family, it has a 4 subfamilies, and these subfamilies are basically defined by the primary amino acid sequence and how these sequences are arranged in specific structures. So there are 4 subfamilies which are CC, second subfamily is CXC, third subfamily is CX3C, and fourth is XC. Okay. Now you can see here we have CXC class of family, where there are about 15 chemokines are there currently. In CC there are around 25, and in XC there are around 2, and CX3C around 1. Okay, now you can see the structural signatures, and CXC there is C, there is X, and then there is C at the specific uh, positions. Then in CC we have C and C at the specific positions, and then in between also we have the CC. In XC we have the C and C at a very different positions, and then CX3C we have C, and then 3X 1 2 3, it can be any amino acid, and then C, and then here it is C. Okay, so it all depends the position of the cysteine residues here. So CC is a class of the family. Like for example, we have CXC, CC, XC, CX3C. This is class. But when we add L as a suffix to a particular class, then it becomes ligand. And when we add R, it becomes receptor. So always remember that in a family, if we are saying it is CCL, it means it is the ligand of the CC family. And when we are saying CCR, it is the receptor for CC family. Okay. So chemokine receptors, they are structurally homologous to the receptors for the hormones adrenaline and glucagon. and they thread through the membrane 7 times. You remember? This is heptahelical, right? So they pass through the membrane 7 times and they transduce the ligand signal via GPCRs. So we call them 7 pass transmembrane receptors. So here you can see this is the chemokine receptor and this is 1, 2, 3, 4, 5, 6 and 7. It has passed 7 times this membrane. So we called as 7 pass transmembrane receptors, and then it is a G protein. Okay, this is, they have an intrinsic ability for the GTP conversion. So many chemokine receptors, they can bind to more than one chemokine from a particular family, and several chemokines can bind to more than one receptors. So this kind of combination is pretty common. Now chemotaxis, if we understand, because these are the chemokine family, chemotaxis is the directed movement, right? Now, and of a cell in response to a chemical gradient, either towards we or away from it. So if the movement is towards the chemical gradient, so you can see here this is the concentration of the chemoattractant. Here it is low and here it is high. So when the cell moves towards it, then we call it as a positive chemotaxis. But when the cell moves away from it, that means from high to low, we call it as a negative chemotaxis. So the chemotaxis or the cellular movement can be positive as well as can be negative. What is haptotaxis? Now haptotaxis is the directional cell movement in response to the concentration of adhesive substrate. For example, ECM. So now you can see here, suppose this is less adhesive surface and this dark green is more adhesive surface. So the cell will move towards more adhesive from less adhesive. So it is the directional cell movement in response to the concentration of adhesive substrates. While in case of chemotaxis, it is not adhesive, rather the the concentration is of the chemoattractant, it is either high or low, but it has nothing to do with the adhesion. So that is the difference between the chemotaxis and the haptotaxis. Now taxis and kinases, these are the two common migratory behaviors which are exhibited by motile cells. So the motile cells, they exhibit two migratory behaviors. One is called as taxis and other is called as kinases. Now let us compare what the difference or what is the comparison between taxis and kinases. So taxis is the direction of cell's motion and is determined by the non-uniform distribution of a physical quantity. That means the cells moves up or down a gradient. That is the direction. While what is kinesis? Kinesis occurs when a cell's speed, it's not direction, it is speed, either translational, that means a single line, or rotational, that means either it is moving around its axis, depends on the spatial distribution of a physical quantity. So here the movement is directional, but here the movement is random, because in kinesis we are concerned about the speed, we are not concerned about what is the direction of the movement. Now, the chemotaxis behavior is the migration up or down a chemical concentration gradient. Then we have aerotaxis, where we see the migration up or down against a oxygen gradient. And then it is phototaxis, where we see the light intensity gradient, and gravitaxis is gravitational potential gradient. So that's how the direction of a particular cell is decided. Okay. Now if we see different type of kinases, then we have the chemokinases, where the speed depends on the chemical concentration. Now here what is important is the speed. So this speed depends on chemical concentration. In orthokinesis, it is the dependence of translational speed or stimulus. So translational means that it is going in a straight direction. And clinokinesis is the dependence of rotational speed or reorientation frequency on stimulus. So that's how we define the two important terms, which are the taxis and the kinesis, and what are the basic or fundamental difference between these two common migratory behaviors. When cells are exposed to chemical gradients, chemokinesis and chemotaxis can both arise. All right. So we can see the directional movement as well as we can see the random movement. We can also see, the speed, how whether it is a low speed or it's a high speed. So in a bounded domain, now it's important. Please pay attention. In a bounded domain with a static concentration gradient, so domain is bounded, that means our area is restricted. Our concentration is constant, it is static, it is not changing. Chemokinesis can result in accumulation of cells. So when we see chemokinesis is what? Cells speed according to the spatial distribution of physical quantity. But here the spatial distribution is static. That means the concentration is uh, static, that is constant. So therefore the cells accumulate due to random variations and their motion direction. So tumbling or rotational Brownian motion takes place. So that's how the cells, when they are moving towards when they are moving in a tissue, and when they found a particular gradient, right, which is static, then the cells can just be there and they stay there for a long time so that they can browse what is happening. So it allows cells to explore their surroundings and causes speed to decrease in certain areas, which is effectively trapping cells there. For example, neutrophils, they swim more slowly in the presence of an immune complex. So when there is an immune complex, there are a number of cytokines, and these cytokines are causing the neutrophils to move slowly, all right, and just be exposed to the immune complex so that it can mount an effective immune response rather than just moving fast and passing through that particular area. Clear? Yeah. Now, if we see the signaling through the chemokine receptors, it can take multiple paths. It can, you can see this is the chemokine and this is the chemokine receptor. So it can go through RAS protein through GPCR and through MAP kinase cascade and AP1. Other pathway is through PCL beta, so it can cause NF kappa B activation. Third pathway is through Rho protein and it can cause cellular movement. And fourth pathway can be through JAK and SLET pathway, and here the JAK through PKC and AKT. It can just either go through survival or through the expression of the particular cytokine gene. Okay, so that's how the chemokine families they work, and uh, uh, they undergo a number of signal transduction processes based on the type of chemokine and the chemokine receptor interaction. Now let us focus about some cytokine antagonists. So cytokine antagonists are the proteins which inhibit the biological activity of the cytokine. So their role is to oppose the activity of the cytokines. They are there to decrease the activity of the cytokines. So they bind to cytokines or to receptors. So basically they block the activity. In either way, they will either bind to the cytokines or they will bind to the receptors and they will block the activity. So let us say this is IL-1 receptor antagonists. So it binds to IL-1R. It's synthesized by the same cells secreting IL-1 alpha and IL-1 beta. So this particular antagonist of IL-1 receptor antagonist, it binds to IL-1 receptor, and it is synthesized by the same cells which secrete IL-1 alpha and IL-1 beta. And inflammation increases synthesis in the liver along with that of IL-1. So when there is a high inflammation, it is not only the IL-1 is synthesized, or side by side the IL-1 are antagonist is also synthesized. The normal function is to modulate the number of receptors that are capable of mounting a physiological response to IL-1. So that's the normal function. So basically modulate the number of receptors that are capable of mounting to physiological response to IL-1. So if this antagonist will block the IL-1 receptor, then definitely the IL-1 cytokine will not bind. And when it will not bind, it is not going to provide any or give any impact. It will not produce the desired effect. So this is antagonist which can decrease the function of the receptor, sorry, the cytokine. The recombinant IL RA, so IL receptor antagonist, RA is receptor antagonist, is used clinically for the treatment of rheumatoid arthritis, right? Now, some natural antagonists are formed by cleavage of the extracellular domain of the cytokine receptor. So here you can see this is the membrane bound receptor, and we have the protease which will cleave this membrane bound receptor, and it will form the soluble receptor. So the free part becomes the soluble receptor. So these soluble receptor components, they compete with the membrane bound receptor for cytokine binding. So you can see here the soluble receptor is binding to the cytokine. For example, IL-2 receptor. So this was one way. Then pathogens can evade cytokine responses by mimicking host molecules and pathways. Some examples we will see where the pathogens, they can just avoid cytokine response by mimicking host molecules and pathways. Now, Epstein-Barr virus, which is EBV, it produces an IL-10 like molecule which is called as viral IL-10 or viral IL-10, VIL-10. It binds to the IL-10 receptor. Okay. Now, when it binds to the IL-10 receptor, then what will happen? IL-10 will not work, and the IL-10 will not be able to mount an antiviral response. Some viruses produce cytokine inhibitors. Okay. For example, the cytokine response modifier CRM protein of cowpox virus. So this is CRM protein of cowpox virus. It inhibits the production of caspase 1 to prevent the processing of IL-1 precursor protein. So no caspase 1, no IL-1 precursor protein, and therefore no activity of IL-1. Some anti-cytokine strategies used by viruses are viral products that interfere with cytokine secretion. Okay. The generation of cytokine homologs that compete with natural cytokines or inhibit antiviral response. Okay. This is also the second strategy. Third is the production of the soluble cytokine binding proteins. So they produce cytokine binding proteins which are soluble in nature. All right, it will also inhibit the cytokine working. Then the generation of viral products that interfere with the intracellular signaling. So that's some viral products, they can interfere with the intracellular signaling, therefore they can stop the cytokine to work. The expression of homologs of cytokine receptors. There are some proteins which are homologous to cytokine receptors, therefore more competition for the cytokine and less chances that the cytokine will bind to the genuine cytokine receptors, and therefore the cytokine will not be able to impart its immunological effect. So that's how these are some of the cytokine strategies which are used by viruses. Now, uh, the last point was the induction of cytokine inhibitors in the host cells. So that means the cytokine inhibitors are induced by viruses, and these cytokine inhibitors will inhibit the cytokine, and therefore the cytokine effect will not be there. Now there are some cytokine related diseases also. Cytokine is not, it's a very good that it controls the immune response, and then it communicates, everything is very good. But then when there is something disturbance in the cytokine related processes or their expression or their function, then that produces very very dreaded diseases. So due to the defects in the complex regulatory network of cytokines and their receptors, worst is the septic shock. Okay. Now septic shock can be due to infection, it can be due to injury, it can be due to trauma, it can be due to cancer, or it can be due to ischemia. In all these cases, there is a overwhelming production of cytokines. And these cytokines are TNF, IL-1, IL-6, MIF, and HMGB1. And they produce the sepsis, okay, or septic shock. Now, the causative agents of bacterial sepsis, the sepsis which is produced by bacteria, are Staphylococcus aureus, E. coli, Klebsiella pneumoniae, Pseudomonas, Enterobacter, and Neisseria meningitidis. So these are some of the causative agents of bacterial sepsis. Then bacterial toxic shocks are caused by superantigens which cause induction of T cell cytokine secretion. So basically superantigens are the molecules which bind to MHC class 2 molecules at their location outside the antigen binding group. So you can see here this is the superantigen and it is binding to MHC, right? And this MHC outside the MHC binding loop. So this is the outside the antigen binding loop. So this is antigen, this is antigen binding loop, but this is binding outside the antigen binding loop. The binding is sufficient to trigger T cell activation. Now, the superantigens can trigger the T cell activations and can simultaneously activate all T cells bearing a particular binding domain and non-specifically activating large number of T cells. Usually, the T cells should be activated only when the peptide, when presented with MHC molecules, should bind to the T cells. But these superantigens, they can cause the wide variety of T cell activation because they bind outside the antigen binding groove and they bind to the MHC. It causes the cytokines to give the same response as they give when they find a particular peptide displayed on the MHC molecule. So that's why we call them as a super antigen. Now, there are several microbial toxins, they act as a super antigens. For example, there are enterotoxins, we have exfoliating toxins, and we have toxic shock syndrome toxin TSST1 from Staphylococcus aureus. Then we have pyrogenic and exotoxins from Streptococcus pyogenes and Mycoplasma arthritis supernatant MAS. So these are all the microbial toxins which can act as a super antigens which can bind to the T cells, all right, and they can just induce the cells to release a number of cytokines and then produce a different disease pictures. So the cause of bacterial toxic shock and food poisoning. So these super antigens, they are the causes of bacterial toxic shock and food poisoning. Now, we have the lymphoid and myeloid cancers. In these lymphoid and myeloid cancers, there are abnormally high level of IL-6 are secreted. And these are secreted by cardiac myxoma, which is a benign heart tumor, in case of myeloma, plasmacytoma cells, as well as cervical and bladder cancer cells. And in myeloma and plasmacytoma, IL-6, they operate in autocrine manner to stimulate cell proliferation, because you know that cancer is what? It is the uncontrolled cell division. And how uncontrolled cell division is taking place, there are so many factors. So one of the factors here is the IL-6, and this IL-6, it operates in an autocrine manner. That means it is secreted by these cells, myeloma and plasmacytoma cells, and it is binding to the same cells to allow them to proliferate more, and that's why producing a very grave picture. Okay. Now, you must have heard this term cytokine storms. So, cytokine storms is a very important term which is used when the production of cytokines or certain cytokines, they are in overwhelming quantities. So some infections, they cause localized exaggerated response resulting in the secretion of extraordinarily high levels of cytokines, and we call it as a cytokine storms. So in lungs, localized swelling, inflammation, and increase in capillary permeability. It leads to pulmonary edema, blocking airways, causing aggravation of symptoms and even death before the cytokine levels can be controlled. So that happens inside the lungs. And it has been found in 1918 Spanish flu pandemic, in 1993 SARS, and in 2019 COVID. So COVID-19. So in all these cases, cytokine storms, it caused a number of deaths. Similarly, in leukocytes associated with a graft. So example, when bone marrow transplant happens, it mount an immune response against the host. So it is all happens because of the cytokine storms. Okay, so we have some cytokine based therapies as well. So cytokines are used for therapeutic purpose like IL-2. It is used in metastatic melanomas and renal cell carcinomas. For example, proleukin is the registered trademark for this. Then we have interferon alpha, IFN alpha. It is used in chronic hepatitis B, C, and certain leukemias and melanoma. So these are the brand names Intron A and Roferon A. Then we have IFN beta for multiple sclerosis by the name Avonex or Rebif. Then we have IL-1RA against rheumatoid arthritis and autoinflammatory syndrome, and the brand name is Anakinra, which is Kineret. Then we have IL-6R, and the clinical use is in cytokine release syndrome or rheumatoid arthritis, and the trade name is tocilizumab, which is Actimra. And then IL-7 for T cell regeneration, post chemotherapy, HIV related immune reconstitution, and this is still investigational, right? So not in the market. Then we have TNF alpha for isolated limb perfusion for sarcoma and melanoma, and it comes under the trade name Biromun. Then we have the GCSF and GMCSF for neutropenia and post chemotherapy issues, and GMCMS CSF for dendritic cell, and it is also used as a vaccine adjuvants. So vaccine adjuvants, they increase the effectiveness of the vaccine, and these are some trade names of GCSF is filgrastim of Neupogen and paclitaxel. And then in CSF, it is sargramostim of leucine. Okay, so this list is just, it shows that cytokines are used for a number of therapies, and they are used in clinical settings, and they have actually helped a number of people to fight with their diseases. So cytokines, and although they are very expensive, but still, people who can afford it, or through the government help, they are very effective in certain treatments. Now we have some limitations also for cytokine based therapies. First is that they interfere with the normal course of immune response. Therefore, they increase the risk of infection and malignancy. Although we treat a particular disease, but you never know that the cytokine may produce some another disease or maybe some malignancy, and this may be an infection also, right? Then, during an immune response, interacting cells may produce extremely high local concentration of cytokines in the vicinity of target cells. but achieving such high concentrations over a clinically significant time period when cytokines must be administered systematically is difficult. We know that this cytokine is there, but then sometimes it is very difficult to reach very high concentrations which are usually achieved in the natural settings, and then the administration of these cytokines is difficult. Many cytokines have very short life, very short half life. For example, recombinant human IL-2, it has a half life of only 7 to 10 minutes. So that means when administered intravenously, we require frequent administration. Why? Because its half life is so short that we again need it inside the body so that it can give the desired effect. All right. So these are some of the limitations of the cytokine based therapies. And then cytokines are extremely potent biological response modifiers, and they can cause unpredictable and undesirable side effects. So even the drugs which we take and the common drugs, they can also produce the side effects, and so the cytokines are. All right. So these are some limitations of the cytokine based therapies. So now let us close our class here and let us summarize what we have learned so far in this class. So we discussed cytokines and their properties. We discussed different families. So we discussed all the 6 families of cytokines, their receptors, their signal transduction mechanisms. Then we focused on antagonists and cytokine related diseases, and then cytokine based therapies. I hope uh, with today's class, although it is a bit complicated and somehow you may feel that the class was a bit very going in a very I would say the a very straight direction. You can again and again see the video, or you can just go through the text to understand more about the cytokines, because ultimately these cytokines are very important for our immune responses. And in all the subsequent classes we will be having and you will be hearing the names of these cytokines and how they are affecting their functions, and how they are causing the cells to perform their immunological functions, right? So in case if you have any feedback, you can always reach out to me at the given email address, and we will see in next class. By the time, take care and thank you.